Hello all. In this video we'll be looking into plant anatomy and plant physiology. This is the first part of the video. Exams Vidhai is a non-profit initiative by the students of IIITDM Kancheepuram to give free coaching and resources to the students who are preparing for national level competitive exams like NTSE and NMMS. So in this particular video we'll be going through the introduction, then the basics of tissue system, and then we'll be looking into the internal structure of root of dicot and monocot plants, and then we'll be looking into the internal structure of stem of the dicot and monocot plants, and then we'll be looking into their differences. So coming to the plant anatomy introduction, ana in Greek language, ana means under, and then temnin means to cut. So it's like cutting a portion and then looking into it. So plant anatomy is the study of internal structure of plants, and atoms are organized into molecules. Molecules are organized into organelles. Then organelles are organized into cells. Then cells form tissues. Tissues form organs, and then organs form organ systems. And then finally, organ system forms an organism. And Nehemiah Grew, an English physician, he is known as the father of anatomy, plant anatomy. So coming to tissues, tissues are a group of cells that are similar or dissimilar in structure and origin, but perform similar functions. So plant tissues are classified into two categories. One is meristematic tissue and the other one is permanent tissue. Meristematic tissues in plants consist of a mass of undifferentiated cells whose main function is to participate in plant growth, whereas permanent tissues, on the other hand, they are differentiated tissues. They carry out their dedicated functions. Tissue system in plants are categorized into three categories. The first one is the dermal tissue system, the second category is the ground tissue system, and the third category is the vascular tissue system. The dermal tissue system mainly consists of epidermis and periderm. And the function of the dermal tissue system is protection of the plant, protection of the inner tissues of the plant, and then prevention of water loss. It has, in the case of leaf, it has stomata, which helps in transpiration, and then in the case of roots, it has root hairs, which helps in absorption of water and minerals. And coming to the ground tissue system, it majorly consists of parenchyma tissues, collenchyma tissues, and sclerenchyma tissues. The function of ground tissue system is help in photosynthesis, storage of food, regeneration of cells, supporting the plant, and then protecting the inner vascular tissue system. And then coming to the vascular tissue system, it consists of xylem tissues and phloem tissues. The major functions of these two vascular system is transport of water and minerals and transport of food. Vascular bundles can further be classified into many categories based on the arrangement of tissues. So the first category is being radial bundles. So here in the radial category, the xylem and phloem are present in different radii and they are alternating with each other. For example, you can take roots. Then for conjoint bundles, which is the second category, the xylem and phloem lie on the same radii and there are different types of conjoint bundles. Coming to the collateral bundle, the xylem lies towards the center, which is xylem is in the center, and then the phloem lies towards the periphery, that is going away from the center towards the end. So then whenever there is a cambium tissue which is present in the collateral bundle, it is called open type. And then whenever, example for this case is dicot stem, and then whenever the collateral bundle is without cambium, it is called closed. And for example for this is a monocot stem. Then coming to bicollateral, in this type of bundle, the phloem is present on both outer and inner side of xylem. The example for this type of arrangement is you can see it in cucurbita. Coming to the concentric bundles, here the xylem completely surrounds the phloem, or the phloem completely surrounds the xylem. So based on the what surrounds what, it can be classified into amphicribal and amphivasal. Then in amphivasal, the xylem surrounds the phloem, and in the case of amphicribal, the phloem surrounds the xylem. Now coming to the internal structure of dicot roots, the outermost layer is called the epiblyma. It is also called the rhizodermis or piliferous layer. Uh, Here the cuticle and stomata are absent. Then it has unicellular root hairs which help in absorption of water and minerals. Then the next layer is the cortex. It is a multi-layered large zone made of thin-walled parenchyma cells with some intercellular spaces. It stores food and water. And the neck coming inside towards the center, the layer after the cortex is called the endodermis. It's the innermost layer of the cortex. Here the cells are barrel shaped, closely packed, and it has show band like thickenings on their radial and their inner tangential walls. These are called the Casparian stripes. But these Casparian stripes are absent in endodermis cell, which are located opposite to the protoxylem. These are thin-walled cells. Then the coming to the innermost uh, class of tissues, they are called stele. All the tissues inside the endodermis constitutes stele. It basically has a pericycle and a vascular bundle. Inner to the endodermis lies a single layer of pericycle. This is the site of origin of lateral roots. And then it has vascular bundle, uh, where the xylem is exarch and tetrarch. The tissue present between xylem and phloem is called the conjunctive tissue. In dicot root, it is made up of parenchyma. And then finally, coming to the center, the last class of tissues are called pith. The pith is present in young roots, but whereas it is absent in the old roots. Now coming to the internal structure of monocot roots. A thin transverse section of monocot root shows the epiblyma, which is the outermost layer of the root. It is made up of single layered thin walled parenchymatous cells. Uh, Again, here the stomata and cuticle are absent. The root hairs helps in the absorption of water and it protects the inner tissues. The next layer is the cortex, which is a multi-layered large zone. It composes of parenchyma cells with intercellular spaces. And then the major function of this cortex is to store water and food material. And next comes the endodermis. It is the innermost layer of the cortex cells, the cortex characteristic cells, which has Casparian stripes and passage cells. The Casparian strips are made up of suberin, which is a chemical, and the last layer is the stele. All the tissues inner to the endodermis constitute the stele. The stele includes pericycle and vascular tissues and a pith. The vascular tissues consist of many patches of xylem and phloem arranged radially. The pith is at the center, and it is made up of parenchymatous cells with intercellular spaces. It contains of abundant amount of starch grains. It stores food. Now coming to the differences between monocot and dicot root. Uh, when you take the number of xylems, in dicot it is tetrarch, whereas in monocot it is polyarch. And then when it comes to the cambium tissues, in dicot root it is present, whereas in monocot root it is absent. Secondary growth stage is present in dicot root, whereas it is absent in monocot root. And then coming to the pith is absent in dicot root, whereas it is present in the monocot root. And then coming to the conjunctive tissues, the parenchyma, the tissues are made up of parenchyma in case of dicot root, whereas the conjunctive tissues are made up of sclerenchyma in case of monocot root. The example for a dicot root is a bean plant, and then for a root of a bean plant, and then the example for a monocot root is the root of a maize plant. Now coming to the internal structure of a dicot stem. A transverse section of a dicot stem reveals, you can see the diagram here. The outermost layer is called the epidermis. It is again made up of single layered cells of parenchyma. The outer wall is covered with cuticle and it is protective in function. The next layer is the cortex, which is divided into three regions, which you can see in the transverse view. The first region is called the hypodermis, which consists of 3 to 6 layers of colon-chyma cells. It gives mechanical support to the plant. Then the next region is called the middle cortex. It is made up of few layers of chlorenchyma cells, which is involved in photosynthesis due to the presence of chloroplasts. And then the innermost layer is called the inner cortex. Innermost layer of the cortex is called the inner cortex, which is made up of layers of parenchyma cells, which is it helps in gaseous exchange and other food storage of food, other food materials. And the next layer to the cortex is called the endodermis. It is the innermost layer of the cortex. It consists of a single layer of barrel-shaped cells. These cells contain starch grains, so it is called the starch sheath. Then the innermost layer of the stem is called the stele. It consists of uh, the central part of the stem, which is inner to the endodermis. It consists of pericycle and vascular. It consists of pericycle, vascular bundle, and pith. The pericycle, it occurs between the vascular bundle and the endodermis. It is multi-layered. It is made up of parenchymatous cell and with alternating patches of sclerenchyma. The vascular bundle are conjoined, collateral, and conjoined, collateral, endarc, and open. They are arranged in the form of a ring around the pith. The pith is the large central parenchymatous zone with intercellular spaces. It helps in storage of food materials. So coming to the internal structure of a monocot stem, uh, the outermost layer is called the epidermis. It is made up of single layer of parenchyma cells. Multicellular hairs are absent, and then the stomata are also less in numbers. The layer next to the epidermis, which is the second layer, it is called the hypodermis. It is made up of few layers of sclerenchyma cells interrupted by chlorenchyma cells. Then the sclerenchyma provides mechanical support to the plant, and then the layer next to the hypodermis is the ground tissue layer. The entire mass of parenchyma cells next to the hypodermis and extending to the center is the ground tissue layer. It is not differentiated into endodermis, cortex, pericycle, and pith. The next layer to the ground tissue layer is called the vascular bundle. Vascular bundles are skull shaped and scattered in the ground tissue. The vascular bundles are conjoined, collateral, endarc, and closed. Each layer of bundle is surrounded by few layers of sclerenchyma cells called the bundle sheath. It has xylems, which consist of metaxylem and protoxylem. The xylem vessels are arranged in V or Y shape. In mature vascular bundle, the lowermost protoxylem disintegrates and forms a cavity. This is called the protoxylem lacuna. And then coming to the phloem, it consists of sieve tube elements and companion cells. The phloem parenchyma and phloem fibers are absent. And then the innermost layer is the pith, which is not differentiated in monocot stems. Now coming to the differences between monocot and dicot stem. In the case of hypodermis, the dicot stem has chlorenchyma cells, and then whereas the monocot stem has sclerenchyma cells. And now coming to the ground tissues, in dicot stem it is differentiated into cortex, endodermis, pericycle, and pith, whereas in monocot stem it is not differentiated. Coming to the vascular bundles, in dicot stem it is less in number, it is uniform in size, they are arranged in a ring, and cambium is present, whereas the bundle sheath is absent. In case of monocot stem, there are large number of vascular bundles. They are smaller near the periphery and bigger near the central. They are scattered and the cambium is absent, but the bundle sheath is present. And coming to the secondary growth stage, it is mostly present in the dicot stem, whereas it is mostly absent in the monocot stem. And then the pith is present in the dicot stem, whereas it is absent in the monocot stem. And the medullary rays are present in the dicot stem, whereas it is absent in the monocot stem. Thank you for attending. Please like, share, and subscribe.